Hi friends, welcome to Foresight, one of the best online coaching institute in the field of IAS preparation. Myself Dr. Vivek Shiva, I am Associate Professor in Science and Technology for this institute. We have been dealing with the subject of science and technology for the past 20 years. So, as far as science and technology is concerned, this is a very important area in UPSC prelims. As you know, prelims is a screening exam and there are 100 questions. 100 questions are asked and these questions are having negative marking also. So once you are committed an error, you have to pay for that. So you have to be very cautious regarding this science and tech area also. Why? You know, in 2014, there were 16 questions from this course. 16 questions means 16% of your chances of your success depended on science and technology. In 2013, there were 19 questions. Your 19% chances of success, science and technology. Similarly, in 2012, again there were 19 questions. And I assure you this trend is going to continue. So you have to be very cautious while reading science and tech, what to read and how to be aware of the current developments in science and technology so that you don't miss the bus. In these 16 questions, there were 11 questions biology and 5 questions physics plus chemistry. In 19 questions, there were 11 questions biology, 7 questions physics plus chemistry. 2012, 9 questions biology and 9 questions physics plus chemistry. This is the picture. Then this science and technology is also related to many areas in ecology and environment. You can't answer many questions of ecology and environment if you have not gone through many areas of life sciences or biology. They are all so much interrelated to each other. They are interdependent. So you have to realize the importance of this interdependence of subjects on each other. So what I want to emphasize, if anybody is neglecting science and technology, he is doing so at its own peril. So friends, let me introduce a brief topic of life sciences, that is blood grouping. So what are blood groups? Blood groups is basically a system of classifying the blood types in various individuals. How we classify them? We classify on the basis of antigens. Now you will ask, what is antigen? Antigen is any substance that triggers a immune response in the body. Triggers immune response. So this antigen can be from the outside. It can be some virus, bacteria, pollen, any foreign material. Or it can be something. This can be out of the body, then within the body. Within means self. So as far as these blood groups are concerned, these are the antigens of the person's own body. They are present on the surface of the blood. Surface of red blood cell. So person is having antigen on the surface of the blood cell and a corresponding antibody against an antigen in the plasma or serum. Let's have a look at the PowerPoint presentation for especially the ABO blood grouping. ABO blood grouping. There are various kinds of blood groups. Say around 35 to 40 blood groups are there. The prominent among them are ABO and RH. On this basis, we classify the blood groups for the purpose of transfusion, for the purpose of paternity testing, and other purposes, say transplantation of organs. So when we are saying this ABO blood grouping, what I mean, this ABO blood group system was discovered by a scientist called Carl Landsteiner in 1900. He got a Nobel Prize for this in 1930s. So as you can have a look in the PowerPoint presentation, there is antigen A present on the surface of the first RBC. It means this person is having blood group A because he has got antigen A on the surface. The second person is having blood group B because he is having antigen B on the surface of the red blood cell. Then you can see of the third picture. Both the antigens are present on the surface of the red blood cell. Both the antigens are present on the surface of the red blood cell. That's why it becomes blood group AB. None of the antigen is present on the fourth red blood cell. That's why it becomes blood group O. Clear? Now we move on to the next PowerPoint presentation. This is about how these blood groups are inherited. You should have a brief idea of Mendelian genetics. These Mendelian genetics say that we derive alleles or factors which are responsible for the expression of these blood groups from our parents. Half of those alleles are derived from our mother and half from father. So if the person is having as you can see in the picture, there is blood group A of the father. So this is OA, this is OB. It means this one allele is from these are the parents of the child. So these parents are having two alleles. One is O, another is A. One is O, another is B. Okay. So when this is say father, this is mother. These alleles they have derived from their father and mother. So when they marry and they produce children, so what will happen? These alleles separate at the time of meiosis or cell division. These alleles segregate first of all. After segregation, then after segregation they independently assort or they can combine randomly with each other. Independently assort. So what will happen? This O can combine with O. This A can combine with O. Okay. This O, if this O combines with O, it forms OO. If this A combines with A, this forms AO. Then there is O, which combines with O. Again it forms OO. Then this forms, then there is B, it forms OB, and it forms AB. So you can see various combinations. This is blood group O, this will form A, this will form again O, this will form B, and this will form AB. So you can have every type of blood group in this. Now let's have a look at the PowerPoint presentation. So what you can see, these antigens on the surface of the red blood cell are determined genetically. We derive them from our parents. Next PowerPoint says what are present in the blood groups. So you can see in the picture 1 there is blood group A. So there are antigens A on the surface. In the group B there is antigen B on the surface. On the group AB there are both antigens. On the group O there are non-antigens. Then there are corresponding antibodies. Antibodies are against the different type of antigen. So the person having blood group A will be having anti-B antibody. The person having blood group B will be having anti-A antibody. The person having blood group AB will be having none of the antibody. So when he is having none of the antibody, he will be a universal recipient, that he can receive blood from anybody, universal recipient, because he is having no antibody. On the other hand, the person having blood group O is having no antigen. So this blood can be given to any individual. So he becomes a universal donor. So these are the whole set of permutations and combinations how the blood groups are decided in an individual. So we will be further taking this to the other systems of blood group in our next class. So presently, if there is any doubt regarding ABO system, blood grouping system, it can be clarified from the faculty. So next time we will be discussing other types of blood group systems and how various kinds of genetically related diseases are transmitted, like there is X-linked recessive disorders like color blindness and hemophilia, how they are transmitted from one generation to another, how the carrier states are determined, how the hemophilic status is determined, how color blindness is determined. We will be looking into those matters. So